Yeah. So here, as we said, the muscle has bundle of muscle fibers. Every muscle fiber considered as a muscle cell, okay? And its multinuclei inside the muscle. You can see the plasma membrane of the muscle fiber, which we called it muscle cell, right? This is the
Right.
plasma membrane, the cell membrane. And inside it, you can see the bundles of myofibrils. Myofibrils, don't forget the names. Type.
Myofibrils.
The myofibrils arranged as a units. Every unit called sarcomere. Okay. What is
Uh-huh.
this between every two lines? This is called sarcomere. This is a unit of myofibril. Okay.
Okay.
What is this? What is this sarcomere? This is a real image for sarcomere. I think you can notice two lines. We call them Z
Mm-hmm.
lines, by the way. And inside or between them, there are thick bands and thin bands. You can see it here. Okay, you can see here.
Actually,
Yeah,
yeah,
thin
thick and then
thin bands here and thick bands here, right?
right.
Yeah, so here thick bands, here thin bands. These are thick bands because there are two types here associated, the
Actin
myosin and
amazing.
actin. Yeah, right. But in this place between the Z line and this thick bands, these are,
Sarcomere.
yeah, these only actins here. Okay. Type.
Okay.
To understand it more, let's focus on this or zoom in on this part and see what are these bands made of. It looks like that. These are the two lines, Z lines here and here, and there are actin. These are actin. The yellow,
Mm-hmm.
uh, the yellow bands are the actins. Okay, actins, actin, actin here and here, and the other Z has also what, actins, but they are not uh, attached. There's space between them. Clear?
They're attached by Dayanin, right?
Now, between the spaces, look, between the actins, there is a myosin protein. This is the myosin.
Isn't it?
The
Yeah.
thick, yeah, the thicker one, myosin. So the thicker band is myosin, okay? And the thinner one is actin. Is it clear?
Yes.
Very good.
Yeah.
So each muscle fiber is itself a bundle of smaller myofibrils arranged longitudinally. The myofibrils are composed to two kinds of myofibril or myofilaments. The thin filament, by the way, the actin is microfilament. You know that, right?
Yeah, it's part of the microskeleton.
Yeah, right. So thin filaments consist of two strands of actin and one strand of regulatory protein. The thick filament is staggered arrays of myosin molecules. We said this, right? The thin one is actin, the thick one is myosin. The skeletal muscle, this is the structure of skeletal muscle, okay? It's called striated muscle. Striated muscle because it's, yani, if you look at the muscle structure, it's look like lines, right?
Nice.
Lines. That's why
Right.
we called it striated muscle. Um, the functional unit of muscle is called sarcomere. We explained it. It's bordered by Z lines, right? Now, by the way, I
Nice.
forgot to tell you here something. This one called M line. M line, the center or the middle between the two Z lines. Okay?
Right.
Mhm.
Yeah, the middle line between the two Z lines.
Do
What's the
This,
do
M line?
uh do both of the sides come towards the M line?
now,
Like when
yeah,
they
yeah,
contract?
we're coming to the technique. Yes. Now, yeah. Let's say you took it, I think. How the
Yeah,
muscle
I think we
contract.
did this.
Let me tell you quickly how it contracts. Now, the muscle contract, look, when we say the muscle contract, it means this muscles becomes shorter or smaller. How it, yeah,
Mm-hmm.
how it becomes shorter or smaller when every
The
microfibril, the microfibrils here, contract. How do microfibril contract? Do you see these Z lines?
yes.
When they come close to each other, and each Z line
Towards the
come
end line.
close to each other here and here and here and here. But how it come close to each other? Look,
Motor proteins.
yeah, there's a theory. The theory says that the actin filament slides over the myosin toward the M line, toward the M line. So this actin and this actin come close to the M line. You understand?
Yes.
It pulls the Z line with it. So that brings the Z lines closer to each other. Do you imagine it?
Yeah.
Yeah, when the Z line and this Z line come closer to
To the
each
M
other,
line.
yeah, toward the M line, that makes the sarcomere shorter or longer?
Shorter.
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 Shorter. When all these sarcomeres get shorter, so what will happen for the single muscle fiber?
Just
Also
smaller.
shorten.
Shorter. When all the bundle of muscle fibers gets shorter, what will happen for the muscle? Gets
Shorten.
shorter.
It just contract.
So this is called
Uh,
summation. Summation.
some.
Yeah.
Oh, okay.
Yeah.
Yes. Yeah.
Yeah. The work of muscle that is a sum of the microfibrils work,
Of the sarcomere.
or this, yeah, the muscle fibers work. You got it?
Right.
The sum of the sum of single muscle fibers work.
Summation.
Hmm.
Summation, right?
It's like what? Depolarization as well.
Yeah, exactly. The idea that many parts work together for one function. Okay, so the overall function or the overall action, which is it, contraction of the muscle, is the or the total of the contraction of the what, the microfibrils. You got it?
My. Yes.
Yeah. Uh, this is called, as we said, a sliding
Sliding filament.
filament model. By the way, we have different models of muscle fiber or muscle work, but this is the most successful model. Okay, this is the most successful model which explain the muscle work in very clear way. Type. So
Mhm.
that is called, we said, a sliding filament model. The filaments slide past each other longitudinally, producing more overlap between thin and thick filaments. But we need to understand how this happens. Look at this. Now, let's understand how this happened. Now, this is the actin. As you know, the Z line is here and here, right?
Right.
Okay. Now, please focus on this point. This is very important thing. This is the myosin, okay? And that is the actin. Actually, the myosins have heads. Do you see these heads?
Right.
Mhm.
Yeah.
Yeah.
These heads
Mm.
has a binding site here on these balls. Do you see these balls?
Yes.
Okay.
Yeah.
This head can bind on a site found on the actin. Okay.
Mm.
Okay.
Okay. So that is called myosin head. Okay. And in the filament, in the actin, the thin one, the actin filament, do you see these openings? These are
Yes.
this
Yeah.
The binding
the
site.
site of binding, the site of binding.
Mm.
Now, what is the idea? That by using ATP, this head bind on the binding site. Okay. By the way, the binding site, we called it myosin binding sites because it's not myosin, it's actin, but it's the binding site of the myosin head. So that's why we called it, this is the binding site of the myosin. You understand?
Okay.
Don't mix in this point. So this is the actin, and the site here is the binding site of the myosin. So we called it myosin binding site. This head of myosin binds on this active site or binding site. Is it clear?
Yes.
Good. But
Yes.
that
Yeah.
that needs what?
Yes.
ATP. That needs energy to be to able to bind.
Biology
Okay.
consideration.
When the head binds on the binding site by using ATP, what will happen? This head bend. How bent? Look at this here, like this way. Look the difference between this point and this point. Do you see?
Yes.
Bent. What will happen when
Hmm.
it bends? It pulls the actin toward
Moves.
left side, right? You got it?
Yeah, it moves
Yeah.
the
Guys,
actin. Wait, the
I,
myosin doesn't move, right? At all. It's just the actin.
no, the myosin fixed, does not move. The
Yeah, and then the act.
actin, yeah, the actin will move. The yellow one will move
Yeah, okay.
according to the bending of myosin head. Look, there are many heads, by the way, not only one head. All
Yeah,
these heads will catch the actin and pull it to the left
a
side.
lot of ADP.
Yeah, this one, yeah, that requires ATP. And this one here, same idea, but toward M line. You understand,
All right.
right?
Yes.
So here, the two Z lines, the two Z lines will come close to the M line due to what? Pulling the actin filament toward M line. You understand? By the
Hmm.
Yes.
heads. Is it clear now for all
Yes, sir.
people? Do you hear me, all of you?
Yeah, so
Yeah.
all like
Yes, sir.
shortening, that's how
Sir.
it contracts.
Yeah,
Yeah.
yeah. This is the most important thing in this lesson today. If you understand it well,
Low energy
so
consumption.
we are done.
Energy
Clear?
consumption.
Time. What is the
Yeah.
business? What is the business of nervous system in this work?
Sends a signal
Helps
to
your muscles when to contract.
To
when
do
to contract.
how the signal will let this. Now the contract, we just understood that the contraction of the muscle is the summation of contraction
Summation.
of the sacromeres, right? Okay,
Right.
Yeah.
now the sacromere contracts in this way. We just understood the technique of sacromere contraction. But the question is, what is the business of the nervous system in this technique?
Probably the binding of ATP,
No,
because
No,
that's
but
something that
you're close to the idea.
um
You think,
release
good,
effect.
good. You think as a designer, if you want to stop this process, what can you do?
Remove ATP.
Think in different way.
Uh, stop
Easy.
it, right?
Yeah.
Just stop the cross bridge.
Yeah, the cross bridge between the head and
um,
the
So
binding
you
side.
stop the binding of the
Yes.
myosin
If
binding
you
sites.
stop the binding between the head and the binding side, this bridge between them, if you stop it, yeah, this one, the crossing bridge. If you stop
Right.
it, so you stop the contraction, right? Or
Mm-hmm.
let's say if it's already stopped, and if you want the muscle to contract, you just open or allow the crossing bridge formation, right?
Right.
That's what the
yeah.
nervous system does actually. The nervous system is stimulate the crossing bridge to be formed. Because once the crossing bridge formed, the contraction happens, right?
Right.
Oh, very nice. So that
Um.
means
So so
hmm,
nervous system has nothing to do with myosin heads being active or being
no,
low
no, no,
energy.
no, 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 no. It just allow crossing bridge to be formed. But here
Oh.
think more deeply. So that means before stimulating the sacromere, the binding site is blocked, supposed to be
Right.
blocked. The
Yeah. Yeah. I
nervous
get that.
system, the nervous system unblock the binding site, so the myosin then able to bind because the energy already present, the head is
Yeah.
already ready, the active site,
So
the binding site already present. So what avoid the binding? Only one thing, that the binding site here is blocked. The binding
right.
site
Yeah.
is, if the binding site is blocked in this state, if the binding site is blocked, so the head cannot bind on it. Here,
Right.
that what is the normally thing. Actually, the binding site is already blocked. Look at this. The
So
binding
most of the
site,
time the
yeah, the binding sites are blocked by this protein called tropomyosin. Do you see this line?
tropomyosin.
Yeah, tropomyosin block the binding sites, so the head of myosins cannot bind on it, cannot form the crossing bridge. Okay, the
Okay.
nervous system or the neurons stimulate
The
the,
tropomyosin.
yeah, tropomyosin to be away from the what, from the
Mm.
binding site.
The
How?
binding site.
Okay, look at the technique, very easy technique.
Just moves it, right?
The nervous system stimulate the fiber of the muscle or the cell of the muscle to release calcium
Calcium ion.
ions. Okay, the calcium ions already stored in the muscle. The
Okay.
calcium, oh, that's why we say always the calcium is important
Yeah,
mineral,
the
yeah,
more I learn
for
about
the
this, is like
muscles
calcium is
work.
very important.
The calcium ions already stored in the muscle. Look here, this is the neuron. This is the
Right.
neuron synapsing with the muscle fiber, the muscle cell. Okay, actually, look what happens when the synapse here, the neurotransmitters. By the way, there's a specific neurotransmitter here is acetylcholine. Okay, acetylcholine
Acetylcholine.
neurotransmitter released from the terminals of the neuron to a receptors on the membrane of the muscle fiber. This is the plasma membrane of the muscle fiber, the muscle cell, you remember? Okay,
Yes.
yeah, that lead to form action potential along the membrane, action potential along the plasma membrane. Then this action potential go through the cell inside, through the fiber, through the T-tube called. This is called T-tube, okay?
So the T tube has nothing else. It's just a
Yeah,
plasma
the
membrane.
T-tube, yeah, is part of plasma membrane that allow the action potential or the impulse to pass through it until it's reach the storage area of the calcium. The calcium is stored in sarcoplasmic reticulum, not endoplasmic reticulum, sarcoplastic reticulum,
Sarcoplastic.
SR you
A.
can say. Another organelle, membranous organelle, such as ER. Okay,
But
similar
it's
to
just
the
for
ER.
storage
Yeah, this one
of
is
calcium.
storing calcium. Yeah, right. Okay, so here when the nervous system, the neuron, stimulates the fiber of muscle or the muscle cell, the SR will release the calcium. Okay, why we need the calcium? Look, the calcium
To,
going to the sarcomeres, go to the sarcomeres and bind on this site. Do you see this site? This is called
yes.
calcium binding site on this protein. I'm coming to, yeah, this protein is called troponin. Okay, so the calcium binds on the troponin. Okay, what will happen when the calcium binds on the troponin? When the calcium binds on the troponin, that change or confirm the tropomyosin, change the shape of tropomyosin, so that that lead to what? To appear the binding
Of the
sites.
Myosin binding site. Yep.
Yeah, when the binding sites appear, so here the myosin head bind on it. Got it?
Yep. So it has these drop-in complexes with calcium
Of course. Okay, we
binding
get.
sites. When it binds, it moves.
Yeah, I will repeat it
Okay.
after we rejoin, please. Yeah, I'm waiting for you. Please quickly.
Okay.
Just one.